for today is microbiome diversity protects against pathogens by nutrient blocking. Fascinating. This recent study is another example of the complexity of the gut microbiota and how we are moving toward a better understanding of the dynamics within this diverse commensal gut community. Very, very popular, very, very important topic. And Dr. Aron, if you wouldn't mind, take it away.
Good morning, Reed.
Sir.
Well, as you know, in each one of these sessions, and we say this all the time, we take a deep dive into recent research. We try to make it as recent as we can that brings to light objective evidence of whatever the topic is. And today, we're going to look much more closely at microbiome diversity and how that protects against pathogens and how it does it through nutrient blocking. And we'll explain that. This study, as well as other recent research, is uncovering the dynamic nature of the microbiome and how the power of commensal bacteria, the good bacteria, become evident only when you see them in, quote, crowds. And so let's look at the actual evidence, Reed, because, and I'll let you say it,
Science matters. So thank you, Dr. Gonshor. After our last broadcast, which was titled Metabolic Efficacy of Time-Restricted Eating, TRE, in Adults. So there were some questions that came to our attention, and one of the questions, Dr. Gonshor, is time-restricted eating effective for weight loss? And again, I'll let you take it away.
Well, thank you. This was only one of a few questions, and I want people to know we really would like questions, and there were a number that came in, so I'm going to be going over a few of them very quickly, because we don't just send an email response to that individual. The idea is we learn together, and so I'm incorporated. So time-restricted eating, TRE, focuses on meal timing. Everybody remember, it's the timing instead of caloric or calorie intake. So a person who is on a TRE plan will only eat during specific hours, and then they fast the rest of the time. So recent studies have shown evidence that it can aid in weight loss, and it may in fact lower the risk of metabolic disease, such as diabetes. So does it work for weight loss? That's one of the questions. So there are some recent studies that show that it has really marginal effectiveness for weight loss. It's not that significant. However, it has also been shown that TRE groups have improved risk factors for metabolic syndrome and heart disease, much more important than the weight loss issue. So recent studies on TRE and type 2 diabetes, used to be called adult diabetes, they have found that TRE offers the benefit of mild weight loss, there is some weight loss, but also much better blood sugar management, which ultimately may be much more important. So recent research then is suggesting that TRE has potential, but not all studies show it is more effective for weight loss than daily regular caloric restriction. So weight loss, it's iffy, but it has some other major issues. It is important, by the way, to note, as science on TRE for weight loss advances, some researchers have expressed the need for caution around who might consider following a TRE protocol. You just don't jump into it. Among people who are overweight or are obese, some studies now have found that weight loss in people doing TRE may be due to a loss of lean mass, that's mass of the muscles, as opposed to fat mass, adipose tissue, fat mass. That's important. That could be dangerous. Therefore, it is especially important for people who are overweight or are categorized as obese and who also have comorbidities such as sarcopenia. And sarcopenia, as many of you might know, stands for the loss of muscle mass. You see that with people who age. Their muscles seem to shrink. That's sarcopenia. And so for people with that, overweight or obese, suffering from sarcopenia, really speak to your healthcare professional before you start trying TRE. There's also a question about and this was actually a question that came in about a week or two ago. TRE's effect on ghrelin. Now, as I've discussed in some of my lectures on intestinal function, ghrelin, remember, is the hunger hormone, and it signals the need for nourishment. So there are data that suggest that intermittent fasting can decrease ghrelin, meaning there's less feeling of hunger. And on top of that, it's coupled with other data that says that there's an increase in leptin. And leptin is the hormone that says, okay, I'm full. So you put those two together, and it's quite powerful. So TRE can have an effect on these two hormones. Lastly, there was the question about drinking outside of the eating window. So typically, the eating window in TRE programs ranges from anywhere from 6 to 12 hours a day, normally 6 to 8, 6 to 10, like that. Outside of that period, a person consumes no calories. However, they are permitted to drink water or no-calorie beverages if they want to remain hydrated, which is important, by the way. In some TRE plans, people may also consume unsweetened coffee or tea with no cream, okay? But that's what they're able to do. So drinking is permitted, and it is important to stay hydrated, because one of the problems with people who go through the various fasting programs is that they become chronically dehydrated. Folks, that ain't a great idea. So much for all of that. There you go. Those are the answers to some of the questions.
Thanks so much. I'd take it a step further, Dr. Gonshor, and say that staying hydrated is required, not just permitted. So if you're doing TRE, you're narrowing the window of your consumption. Dr. Gonshor said 6, 8, 10 hours a day. It depends on the person and the type of program or what your goals are and things like that. But you're going to restrict your eating to a certain part of the day, but not your hydration. That's really kind of a must. There's lots of other things and questions that might come up, and we'll continue with this discussion. But for right now, I want to just thank you for submitting those questions. They're really, really important. It gives us some fine tuning. We all get to learn together. So now let's jump into today's study about microbiome diversity protects against pathogens by nutrient blocking. Dr. Gonshor.
Thank you, Reed. Well, let's take a look at this and see where it takes us. So I'm just going to, there we go. So microbiome diversity. What's the story with nutrient blocking? Well, this is a study out of the UK, out of England, and a very, very well-written study. And what they're saying is that, and I've put this in bold, diverse communities of resident commensal gut bacteria, the good bacteria, collectively protect the human gut from disease-causing microorganisms. And how they do that? By consuming the nutrients that the pathogen, those pathogenic bacteria, would need to be able to gain some kind of a foothold in the host. So what does that mean? Well, this is a new study in Science. And what they're saying is that when you have a large, diverse community of gut bacteria, they compete with pathogens for nutrients. And the pathogens may not have enough food fuel to colonize and invade the body. So this competition protects the gut against invading pathogens. And this particular type of action has been coined as colonization resistance. It's the colony that creates the resistance. So the more microbes there are, and the greater that diversity, the greater the variety of nutrients that they're likely to eat. Because the more bacteria and the different kinds of bacteria they will eat different types of things. This increases the chances of a nutrient overlap with the pathogen, because at least some of these commensal bacteria are going to be eating the same stuff. So the greater the overlap, the better the host is protected. So let's delve into this more. So in the study, they asked how and which gut microbiota protect against microbial pathogens. And in the methodology, what they did is they tested about 100 common gut symbionts. Symbionts are species living in the gut in symbiosis. They're living in a very, very homogeneous way with the body, not harming each other. That's a symbiotic relationship. And they studied these 100 common gut bacteria individually, and when they were in combination of increasing diversity with a whole bunch of other bacteria. And they wanted to look at the ability of all of that to limit the growth specifically of two bacterial pathogens, Klebsiella pneumoniae and Salmonella typhimurium. Tough to say that 10 times quickly. And the results that they found first in germ-free mice, and then they did it in humans, what did they find? Interesting. Individual bacteria, whatever they were very poor in their ability to restrict the spread of either of these two pathogens, the Klebsiella and the Salmonella. But when they were combined, especially diverse groups of up to 50 species, when they were cultured together, the pathogens grew, listen to this, 1,000 times less effectively than when they were cultured with just one species, whatever that species was. And when you had those 50 bacteria together and they included E. coli, the effect was even better. Very fascinating. So it brings up this issue of nutrient blocking, because both in vitro and in germ-free mice, you require a diverse range of human gut bacteria that are going to be able to consume nutrients that a pathogen needs to grow. So you're competing for those nutrients. And consequently, what happens when you have that diverse group, it blocks the pathogen from invading the microbiome. So findings, you need ecological diversity. That is the important thing for colonization resistance. And secondly, the community composition of all these bacteria is important. There are certain combinations of what they call essential key species that are needed within these diverse communities, since these key species increase the overlap between the nutrient use of the community and a pathogen. So put another way, these key species have a particular way of being able to block the effect of the pathogen, because certain microbiome species are found to be essential for community-based protection, even though these species offer little protection on their own. If you see them on their own, they're not doing very much. Put them in that group, magic begins to happen. So these communities are blocking the pathogen's growth. How? By consuming nutrients that would be required by the pathogen. And the concept of nutrient blocking can be used to predict what specific sets of these commensal microbiota that will resist a novel bacterial pathogen. And how would you do that? By using genome sequence data alone. So that's very interesting, because if you're trying to build a better mousetrap, you'd say through genome sequencing, what are the groups of bacteria that we need that are going to compete best against those pathogens by competing for the nutrients? So I'm quoting from the study, despite this complexity, these ecological patterns are actually explained by a simple underlying principle. And what's that principle? The collective ability of certain communities to consume nutrients and block pathogen growth. Nutrient blocking effect is a property of the entire community. rather than of any one species alone. That's very important. The fact that colonization resistance is a community level trait explains the importance of the ecological diversity and complexity that we observed in our experiments. So in addition, the most protective communities were composed of species with highly similar protein compositions to the pathogenic species, and they provided the best colonization resistance. Not surprising, because if you have commensal bacteria that look very much like pathogenic species in terms of their genomes and so on, they are going to compete for the food, and those pathogens are not going to be able to get to it. So colonization resistance is only observed once communities share sufficiently high overlap in their carbon source utilization profile with a pathogen. Moreover, communities with the greatest metabolic overlap with a pathogen provide the greatest colonization resistance. When you think about it, it makes a lot of sense, but you got to prove it, and they seem to have. So when tested experimentally, the communities with the highest nutrient overlap were up to 100-fold more effective at reducing the pathogen's abundance than communities predicted to give weak protection. This is actually quite fascinating. So the takeaways: nutrient blocking. Remember, diverse range of human gut bacteria consume nutrients that a pathogen needs to grow. And by doing that, it blocks the pathogen from invading the microbiome. The most protective communities of bacteria are composed of those species with highly similar protein compositions to pathogenic species. They look like the pathogens, but they're not. They're commensals, and they provide the best colonization resistance. And the findings highlight that microbiome diversity is important to human health, as is the key overlap in nutrient utilization profiles between the community and the pathogen. So this points to the development of new strategies for optimizing gut health, and for, frankly, the rationale of design of pathogen-resistant microbiome communities. And with that, it's the end of today's Science Matters.
Thank you, Dr. Gonshor. That was a good presentation. Kind of backs up what we've been saying. And I've used for 20 years the term crowd out. We want to crowd out the bad. So it's not just about identifying a pathogen and killing it, because you might kill a lot of friendly things in fire. And so crowding out these unwanted or unfriendly bacteria would be a better strategy. And this study takes that to another level, which I really appreciate. So if you've enjoyed this at all, please subscribe, like, and share, and continue to submit your questions, comments, and suggested topics at www.fdntraining.com/science matters. And if you'd like to look at the past recordings and also get copies of the studies, go to fdntraining.com/studies. And join us again in 2 weeks when we'll talk about dose of jogging and long-term mortality, because
science matters.